Okay, our research focuses on creating immunotherapies for treatment of methamphetamine abuse. And there are no FDA-approved treatments right now to treat methamphetamine. And a project that's been going on a long time has been the development of immunotherapies to treat methamphetamine abuse. And that started about 15 years ago, and I joined the project in midstream. My unique take on the project is to reformulate these antibodies, put them into gene therapy constructs, and deliver them to patients. Now, the way that works, the way that antibodies work to treat methamphetamine abuse, is that unlike small molecule compounds that cross the blood-brain barrier, they bind to receptors and they either block the effects of meth, or ideally they would. There's nothing out there like that. They would block the effects of meth, or they would substitute the effects of methamphetamine, much the way that some opioid treatments work. The way the antibodies work is they stay in the bloodstream. They don't cross the blood-brain barrier, and when someone takes the drug, the antibody binds to the drug itself rather than blocking the receptor. And so in that way, they don't have any addiction potential, and they blunt the effects, the rewarding effects of the drug. And so what happens is we imagine someone would be in behavioral therapy to stop using, these medicines won't be used alone. We see them in behavioral therapy. They have the antibody on board, and if they slip, if they take the drug, the antibody will be there to protect them. And so it'll blunt that, and it'll give them another chance, a second chance to get back on board and stay drug-free. And one of the challenges of these antibody therapies is having one that lasts a long time. One of the problems with drug addicts is that they have a problem with patient compliance. So what we sought to do is to create a medication that would be extremely long-lasting, that they could have one dose and it would last for a long time, for the months that it might take them to actually change their behavior. So we're at the very first stage of that. And the first stage of that is that we've packaged DNA that codes for an anti-methamphetamine antibody fragment. And that is encapsulated into a non-replicating virus, the relatively safe virus. And this is not a novel approach for therapies in general. This approach is being studied to treat hemophilia, Lorenzo-Oils disease, adrenoleukodystrophy, and others. There's several others. There's a recent Forbes article on this approach, as well as others that are looking into possibly using this as a treatment for nicotine, as well as cocaine addiction. So these were all kind of being developed around the same time, and our approach is with methamphetamine, and we have the methamphetamine antibodies. So package all of that into a non-replicating virus particle, and then one injection into the patient induces a cellular response from the DNA that they create their own antibodies. So it's sort of like vaccination, except instead of being vaccinated and you have variability in titers, they're producing all one single kind of high affinity antibody fragment. And we know the affinity, it's been well characterized, and this is a fragment of a monoclonal antibody that's actually in phase 1B trials right now. So this is the next generation, is what we imagine. So what we did is we packaged this. We had two different antibodies that we packaged into viral particles, and we injected those into mice one time. And we did a pharmacokinetic study 50 days later. This is 50 days after the initial injection. And what we did is we injected the mice with 1 milligram per kilogram of methamphetamine, and then we took blood samples to see what the levels of methamphetamine were in the blood. And what we found is there was a significant difference in those animals that weren't treated and those animals that were treated. And so that's the simple explanation of what we have so far. I think the next stage of the research, because these are early stage of the experimental medications, is first of all, they need to be safe. We can't go forward with these medications if they're not safe. So we will study safety, we'll study the response, the immunogenic response that the animals might have to making the protein. also any liver function issues, a lot of those things that need to be explored first. And at the same time, we want to explore further efficacy. And there are a lot of different things that we need to study in order to see if these are effective medications. One is self-administration models, whether animals that are taking methamphetamine will stop self-administering methamphetamine after getting this treatment, as well as other animal models of addiction to show that they're efficacious. That's where we envision that this medication can help, something that will give them protection that can help them make better choices and protect them if they slip. It has a personal element for me, being from a rural background in Missouri and seeing methamphetamine take hold in some of the areas that I grew up and love. But I love the idea of making a medication, and that's what keeps me going. I started attending AAPS meetings probably in 2009, I think, or maybe 2008, even earlier, and just fell in love with the diversity that AAPS offers, because we have a great mix of pharmacological professionals, pharmacy professionals, pharmaceutical sciences, as well as academics. So there's a nice balance between discovery and the companies that could actually bring a drug to market. So as someone in translational science, science that we want to develop into drugs, it's a great opportunity for me to see and to learn from all the talks and all the poster presentations what aspects are important in making a good drug. What is drug like? And that's something I've learned. And I've learned from being involved in AAPS.